In 1513, Spanish explorer Juan Ponce de Leon led an expedition to Florida in search for the Fountain of Youth. He never found this fountain, but if he did, would he be walking around today, 506 years later, with silver hair to the ground, wrinkled skin, and a rounded back? Would he have cataracts, arthritis, and osteoporosis? Having drank from the Fountain of Youth, would he have the ability to live forever but keep aging? And what is aging? We all recognize the signs of aging, but at the molecular level, no one knows what causes it. The answer may lie within our mitochondria, familiar to us through its role in energy production. Less familiar to us is the mitochondrial genome, which codes for the machinery needed for energy production. Interestingly, mitochondrial DNA is damaged more rapidly than nuclear DNA, and mitochondrial DNA damage accumulates over the course of a lifetime. There's a correlation between this accumulation and the onset of aging-associated pathologies like cancer, diabetes, and neurodegeneration. The mitochondrial hypothesis of aging predicts that mitochondrial DNA damage accumulation leads to cellular dysfunction, which drives these pathologies. What's unknown, however, is whether this connection between DNA damage and the onset of these pathologies is correlative or causative. In order to effectively treat these pathologies, it's critical to understand this, and that is the goal of my research. I'm going to challenge the mitochondrial hypothesis of aging by identifying the key players that are involved in maintaining mitochondrial genome stability and testing whether the manipulation or presence or absence of these genes can affect aging in mice. Our lab has developed a system in which for the first time we can study DNA repair in isolated, intact mitochondria in a system free from nuclear effects. I've started these studies in healthy, unmodified cells, then we'll genetically manipulate or pharmacologically treat them to assess the need for various genes in DNA repair. I'll then move on to mice, in which I will manipulate the genes that I found to be necessary for repair in mitochondria and assess the need for these in mitochondria and test whether this manipulation will slow down or speed up aging in mice. Ponce de Leon never found the Fountain of Youth because there is no Fountain of Youth. The mitochondrial hypothesis of aging suggests that there is a fountain of aging, which is the cascade of effects caused by the accumulation of mitochondrial DNA damage. I'm going to challenge this hypothesis by identifying the key players responsible for maintaining mitochondrial genome stability and whether the presence or absence of these genes can elicit or delay the effects of aging in mice. The potential of this work is insight into one of the most fundamental and unanswered questions in biology. What is the molecular basis of aging? In an aging world population in which the incidence of age-related diseases is rising, my examination of the fountain of aging may drive the development of therapeutics that can treat or delay many age-related diseases. Thank you.